All right, hi, yeah. Snow day, 2 days. That's fine. I'm gonna go over the review real quick, but I know the real reason you're here is to see my cat. Oh, we got Ike. Oh yes, he's wearing a bow tie. Look how cute he is. He's not happy. No, he's not. It's okay. See, I could hold him forever. He's actually a really good cat. Okay, he's done. All right, so we left off with meiosis. These notes are online, and you can go through them along with it if you want. So we finished off mitosis. Meiosis is pretty much the same thing to start off with. So you're going to go through your interphase G1S, G2. So you're going to double your DNA. And then the first half meiosis, meiosis 1, the key thing is where the homologous chromosomes line up next to each other. So in prophase 1, that's where your tetrads form and you get your crossing over. And then you'll do metaphase where the chromosomes will line up together. Now in the middle, so two number ones, two number twos, two number threes. And then in anaphase, the homologous chromosomes will separate. And then in telophase, you'll reform chromatin, just like in mitosis, and separate to form two cells. These are now haploid, even though they have double chromosomes. So the first thing we have to do next is separate those double chromosomes into chromatids. So what's happening in meiosis 2, you're pretty much just right mitosis, but with half as many chromosomes. So all the X's are going to reform, they're going to line up down the middle, the sister chromatids are going to separate, and then you do telophase and cytokinesis. So what do we end up with? 4 haploid unique cells. So because of the crossing over, each combination is going to be completely different compared to the others. So how many options can you get? Well, it depends on how many chromosome pairs you have. So if you look up here, we have 3 chromosomes in that cell. Well, originally over here we had 6 chromosomes, so 3 pairs. So you would do 2 to the 3 power, which would be 8. So down here I have 10 chromosomes. That would be 5 pairs, 2 to the 5. There'd be 32 different egg or sperm combinations. There's a little chart about comparing mitosis and meiosis. You could even do this if you want, but there's some of the key differences between them. You could also talk about how homologous chromosomes line up more in meiosis and the sister chromatids separate in mitosis. All right, so last thing is our signal transduction pathways. So you should know the 3 key sets of reception, where the ligand will bind to the receptor, and that will cause a conformational change in that protein, which will activate something on the other side of the membrane, unless the ligand, of course, can go through the membrane and find a receptor inside. Transductions, and we pass that signal along from one protein to the next. And then finally, a response will happen. So either something will go into the nucleus and change DNA expression, or maybe you'll activate a protein in the cytoplasm, or you'll signal a protein to get destroyed. So any of those things could happen. So you should know some of the receptors in general. So G-coupled receptor proteins, so the receptor that will couple with the G protein and activate it by combining it with a GTP, and that will then move to a different protein and activate it when they combine together. And so those are the G-coupled. So the receptor, coupler, the G protein, and another protein is involved. The RTK proteins that will be phosphorylated, and they can activate other proteins, and then ion channels can let ions in, which can also alter cell function. But remember, you have to pump those back out in order to reset it. And then finally, intracellular, so small nonpolar ligands like steroids can go through the membrane, and they can bind inside of the cytoplasm. So for transduction, 2 things we talked about were kinases and phosphorylation. So a kinase will phosphorylate a protein and activate it, which then is likely going to be a kinase and activate something else, and we call that a cascade. And then you also can use second messengers to transduct and amplify a message. So you could use calcium or that CAMP that we talked about. So what's the good thing about a second messenger is, one, those messengers can sometimes activate multiple pathways, so you can do multiple responses to one message, and also you amplify it. So every step you go down this pathway, you're going to amplify it 10 times possibly, and then it'll make the message much stronger from what it started out as. Finally, your response again, you can go into the DNA and activate new sections or transcribe new RNA to make new proteins, or you can possibly activate or inactivate proteins that are already there or mark them for destruction. Last thing is feedback loops. So again, positive feedback loops. Whatever happens is going to happen more. So we talked about things like birth, contractions, or milk production. And then negative feedback loops is whatever happens less. So the stimulation causes it to go the other way. So things like maintaining your body temperature, your blood glucose. If your blood glucose increases, eventually your body's going to cause it to decrease to try and maintain balance. So if you have any questions or you need anything, you can email me. Yeah, bye.